This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
Hey everybody, it's Razib Khan here with the Unsupervised Learning podcast, and I am here today with soon-to-be Dr. Daniel Tabin. Daniel is in David Reich's lab. Could you tell us a little bit more about yourself and your research, Daniel?
Yeah, well, we'll see. Hopefully soon to be, and not that soon. But yeah, I'm a fifth-year grad student, and like David, I study ancient DNA, and sort of my thesis will be on the history of Central Asia and Siberia. That's sort of the thing that connects the main things I've been interested in. And although I've done other things, including recent-ish, like last year paper that's sort of a technical comment challenging another paper on ancient China.
Okay. Well, so also I will recommend for the listeners out there, check out Daniel on Twitter. I'll put the link in there. Really good science communicator. He has not fled to Blue Sky. There are it's a lot of science Twitter has kind of like disappeared, and you're one of the people that I still see in the genetic space. You, maybe Sasha Gusa, a few other people. So I wanted first to talk about something technical, then maybe we'll get to ancient DNA, maybe some of your other research and the results section, so to speak. So the paper I want to talk about is more of a comment section, not a method section paper, right? Concern about ancient DNA sequences reported from late Pleistocene individual in Southeast Asia. And so that came out last year. And I'm assuming you've been looking at that longer than 2025. And it's about the Red Deer Cave, which I've heard about. That's in like South China, right?
Yeah, it's I guess somewhat famous because, and this probably prompted at least some of the research on the sample. It's like the skulls look very interesting phenotypically, and they were even potentially thought to be kind of archaic hybrids before the like ancient DNA revolution, excuse me. I started looking at actually in 2020. I read the paper, which made some very interesting claims, and I sort of wanted to like forward them. I thought this may be related to the population Y signal. For context, I should say maybe that the population Y signal is idea that certain Native Americans, specifically those in Brazil, have some sort of affinity to certain Southeast Asian groups like the Onge, sort of Andaman Islanders, and potentially Papuans. And there's the MZR paper suggests that there was a migration from Southeast Asia into Northeast Asia and eventually reaching the Americas. And I looked at the paper and I basically, I guess, sort of to spoil it, I realized that the claims didn't really make any sense, and kind of more concerningly even than the claims not really being super substantiated was that the data was like highly problematic in sort of a way that sort of in both ways that are sort of typical and ways that are atypical, I guess. So I then tried to figure out what happened. I also want to say, before I say anything, I want to make sure I get this out, that the authors of that paper are just extremely kind and actually like very, I would say, collaborative in some way. I mean, they didn't like directly contribute to the notes, but like I sent them the notes, and I felt that they were, even though they didn't necessarily say, oh, we're 100% wrong at any point. They like gave feedback very friendly, and I've heard of other stories where people are trying to critique science, and people are very aggressive and rude and mean, and they were very gracious. And I wanted to actually shout them out for, even though I disagree with their conclusions. And I think that the data is problematic, and I think it shouldn't be used. I actually think the authors are wonderful people, and I wanted to make sure I said that.
Yeah, I mean, that's how science should be done. But let's be candid, that's often not how it is done. People get very attached to their results, especially for cool results. I'm going to tell a real quick story, because this is like it's a long time ago and people aren't going to know who this is. When I was in graduate school, I had a close friend who was in a lab, and it was I'm going to just say it's a plant genetics lab. It's a prominent one. And a few people are going to be able to guess who this is. But the TLDR is my friend was a first-year graduate student in the lab, and he could not replicate a science publication from like 2 years before I think he joined the lab, and he became convinced that it was fraud. And they didn't explicitly ever say it was fraud, but the primary investigator, the professor, eventually did withdraw this science publication. Like, it's a science publication, like that's tough, right? Um, and so from the outside, it's like, okay, this is how it should have worked. But I will tell you, my friend had to drag the professor to that retraction. Okay, he threatened to quit the lab because he was not going to work on a project where he knew that the previous stuff he would have to cite was probably fraudulent,
Oh.
And so, if when you see how the sausage is made, it's tough. It's a human enterprise, and so we definitely, should give a shout out to people who are collaborative in this way. And Daniel, data submitted upon request is often more theoretical than reality. I've seen people who've actually tried to do this, and half the time the data is just not given to them.
Yeah, that's actually a great example from this exact situation. Like, again, while they didn't necessarily accept the claims in, they actually kind of wrote a rebuttal to my paper, which people should read. I disagree with it, obviously, but people should read the rebuttal they wrote to my rebuttal in some way. But actually, like, they gave me the FASTQ files. Like they don't. The FASTQ files weren't published, just like the BAM, like that data's available to anyone. But I actually have the FASTQs, like the original reads, which is like extremely generous of them. And like, if you're like going to war with someone, you're not going to give. They were essentially giving me more ammo in some way.
Yes, they were
extremely gracious of them.
They were letting you take them down. It doesn't sound like they slow walked it. They made it easy for you to make your case, even if they disagreed. That's how it should be done.
I think that they disagreed, but I think that they did want to. They did continue to agree with their conclusions, but they ultimately were also in favor of the process, and I strongly commend them, because I've seen it myself, not in personal experiences, but like people I know where, like, yeah, you're totally right that academics are often very want to admit they're wrong, unfortunately.
Hey, they're human. They're human. So going to this paper and this whole issue of contamination. So you said FASTQ files. Some of the listeners of this podcast are actually biologists, so they'll know what that means. So just to be clear to the listener, when you have whole genome sequences, you get these raw reads, right? These FASTQ files, and then you take the FASTQ files, you reassemble them into something called like a BAM, or now a CRAM file, I guess, which is condenses it and kind of cleans it up. And there's like data, deduplication, all of these things that you do in the FASTQ files. And then you take the BAM and you often reduce that down to just the variant, so like the VCF file, right? Variant caller format. And from that, you often, like, in your lab, David Reich's lab, often use SNP arrays, which are somewhat different. And those are just the single nucleotide, variants like that are on an array on a DNA chip. And so, when I write about ancient DNA, mostly I'm writing about the results, and, I'm interpreting through the historical lens and whatever. But there's a lot of technical detail on how we get that data. And some of these results and some of these write-ups that I'm doing are premised on the idea that the data is actually correct, but you're not doing like you're not doing analysis of contemporary data. You're doing analysis of DNA of genome sequences, or doing the SNP capture array, thousands of, sometimes 10,000 years old. I think this is 14,000 years old, this Red Deer Cave sample that you're talking about. Can you tell the listener, in ways they could understand, how you figure out that this is, one, the data from the individual in question, and two, I'm assuming, I know there is, damage and degradation, how you clean that up so you can compare it to modern genome sample?
Yeah, those are great questions. Yeah, no, it's and I'll say, kind of as a side, MZR as a process, like the project, actually was a brutal awakening for me, that like the data in general should not be blindly trusted. And yeah, I started it my first year, and it was sort of the some light in my eyes was replaced by dark cynicism. But no, it's true. It's like people do their best, but like it's really hard to get these DNA out of these samples. It's really, really difficult, and many of them fail. Anyway, so to answer your question more directly, I guess the first thing you do is dating the sample, which is actually, I think, an underrated but super important thing, because while archaeologists are very often totally correct about their assessment, there can be things like where the sample comes from. There can be things like a rodent who moves the bones around, or like just like it gets mixed up in a drawer because it was actually excavated in the 1700s, and at some point in the last 300 years it got mislabeled or something. A kind of interesting example, which I'm not sure many people know about. I'm curious if you know about it. If you do, I apologize for repeating it maybe. But the Cro-Magnon man, like the Cro-Magnon man, like the original one, at least part of its skull, it seems like its skull is a little bit of a mishmash of like some very ancient person and some more recent people. Potentially someone was like, oh, there's a bone missing, it's this bone, and like kind of filled it in. I don't know the exact details. Anyway, that bone actually was dated. It's the only bone of the Cro-Magnon man we got DNA from, because it's the most recent bone, and it's like medieval basically. And so I think that's published in like Current Biology, some paper. It's like buried in a supplement. It's not particularly interesting, but it's a little bit sad that like the Cro-Magnon man, which like Cro-Magnon, that's like the name given for cavemen for it's sort of not actually maybe even from that period. So the first thing you do is dating, usually with carbon dating, but there's other methods. And then once you have the data, you can look at a bunch of metrics. For example, the amount of damage, just in the entire library. Which the library, I should clarify, maybe is like the BAM you get from the data. So you it's actually like a physical thing, but you make some physical kind of soup of DNA and kind of biochemically in ways I'm going to kind of just ignore, and then you sequence it, and you get this FASTQ files you're mentioning, and you align to get the BAM. Anyway, so that once you have that, you can look at the rate of mismatching to like the human reference genome. And of course, we're not all clones, so there'll be mismatches all over the place. Although actually, the amount of human differences per base is like 1 in 1,000, so they're not that common. But you want to see that there's an excess of C to T kind of almost like mutations, not mutations. They're like implicitly not. They're kind of errors in some way, or like modifications that happen to the DNA through time.
Yeah, and just to be clear, we have A, C, T, and G for the DNA. That's what you're talking about.
Yeah, sorry. Yeah, and there's a thing called the process called deamination, which I believe, I'm not a biochemist, I'm really like a fake biologist, my background is math, computer science, that there's this amylase that like gets removed from the cytosine and actually turns it into a uracil, which people may not be familiar with is a U, and it's the RNA version of a T. They're like RNA has like I think that's the main difference, or maybe some other differences. Again, I'm not actually a real biologist, but like RNA has a U instead of a T. And so when you read it with like a DNA sequencer, it reads as a T because it's like quite similar. And so these cytosines, the Cs, get turned into really uracils, which is the U, but it's read as a T. And you can look at like the damage rate overall, and that should, in theory, be elevated compared to modern DNA. And like it should specifically be the C to T damage. And then additionally, you want to see it on the ends of the reads, because the ends of the reads sort of get frayed. You can imagine they're kind of like getting broken apart. And so you should look at the ends of the reads. And if you have what's called a single-stranded library, which I don't want to get too technical here, but basically, and also this is not my expertise either. Basically, if you read in like sort of just like the one half of the DNA, you'll get the C to T on both ends because they're kind of frayed on both ends. But if you do what's called double-stranded library prep, where you actually bond it to like kind of novel DNA on the other side to kind of make like a mirror copy, then on the otother end of on the it's called the 5' end, I believe, you'll still get the C to T damage. You actually get the reverse on the 3' end, which is G to A, which because G in like DNA is always kind of like attached to a C, and T is always attached to an A. So the C's turn into T's, but that actually just makes G's turn into A's. I hope that's not too technical. And that's kind of the first thing you can do. And there's other things as well to look at contamination, which I can I like to talk about like looking at the MT and the X chromosome, but I'll wait there in case you want to jump in.
No. I mean, so you were saying you're a fake biologist, which some of the listeners would probably be a little confused or laughing at that. This is not an uncommon thing. I will tell you a story. Do you know Michael Torelli at UC Davis, quantitative, evolutionary biologist?
It sounds familiar, but I
Okay. So anyway, so Torelli, one, his writing is illegible. That he's famous for that. He was, he was Felsenstein's postdoc, and I remember one time I was in a seminar with Torelli, and he was explaining about recombination rates and how they decrease at the telomeres and they're maximized at the centromeres. And all of us who had taken the recent molecular genetics course looked at each other because in humans, it's the opposite. And so Torelli is he's the National Academy of Sciences. He's a very, very prominent quantitative geneticist, but, biology is a massive field and no one is going to be the specialist or no one's going to know, very much of it, to be candid, right?
Yeah, no, it's true.
And so,
I think it's also true of other, the well of knowledge is so deep that if you want to become specialized in knot theory or something in some obscure field of mathematics, you're going to not know much about. You'll know much more than the average person on the street, but you won't know much compared to your fellow experts at other places. I just feel like our field specifically. I mean, you might relate. Maybe you don't, but I sort of feel like a jack of all trades, like a master of none. I potentially know far more math and computer science than some other people in who are doing real biology, but then I know so much less than people in math and computer science programs. And correspondingly, I know so much more biology than my friends who are doing math and computer science, but so much less computer science and math than them.
Well, one thing that I will say, in discussions, this meta question here, I think part of the things in genomics is the first human genome the draft was completed 25 years ago, probably around when you were born, right? So it's a very, very young field, and it's moving extremely fast. And so, you could be really good at doing something, and then it's totally irrelevant. So, I think I've been out of academia for a little while, with RNA-seq and, understanding gene expression with RNA, sequencing, for example. I know when I was in grad school, some of the younger grad students, they didn't know what gene expression arrays were. Do you know what they are?
Only vaguely.
Okay, well, they were, again, older biologists are going to be laughing because they were very, very cutting edge in 2005. Okay.
Wow.
But they just kind of got phased out about 5 years later when whole genome sequencing came in. So gene expression arrays just were the way you figured out which genes were being expressed in what tissues and stuff. They didn't do RNA sequencing, right? And so there were people they knew how to read these arrays, how to analyze these arrays. There were whole pipelines for the arrays, to understand them, and it was just totally irrelevant within 5 years. And so I think genomics is special that way in terms of you're always having to update and you're always having to accept that you don't totally understand what's going on. And, we're talking about contamination here. I'll give you a quick story that I've heard many times from many different people. But initially, geneticists were not aware of artifacts introduced by the sequencing technology you used. And so there was a period where I don't think it was ever published, but I think some things like this have been published. But basically, someone created a phylogeny of a population. So it's within a single species. And they found this novel, crazy structure that they had never expected, relationships that were never expected. Right before publication, someone looked at the methods and they matched the IDs of the individuals to the sequencing platform. Because this was in a period where sequencing was extremely expensive. It was very bespoke, and not many labs really had access to cores. And it turned out the phylogeny was just a recapitulation of the sequencing technology.
Yeah, that's funny.
Because the errors within each sequencing technology were different from the errors in other sequencing technology, and so it was a phylogeny of errors, right? And so obviously today that wouldn't happen, partly because sequencing is so cheap and it's so standardized and so but 10-15 years, 15 years ago this was still happening, right? And so
all ways that I guess I said before, you don't trust the data fully, but I think to some degree there still is evidence of these things. I know that someone else in the lab, a postdoc named Roman Ferrier, just published a preprint on a set of SNPs that are more compatible across 1240K twist and shotgun, because it's not huge, but you do get sort of these weird things where you get individual one shotgun individual to 1240K. For people who don't know, that's
Yeah.
like a different type of that's like different types of sequencing. I mean, you like
Yeah.
compare the two individuals, like you have two individuals, the same individual, but one's shotgun and one's 1240K. So it's 4 fake individuals, but it's really just 2. And actually, you see an attraction between the different sequencing profiles.
Yeah.
And like I know also in our lab, when you do what's called like variant calling, which is where again you have all these different reads of DNA, and you kind of choose, okay, what's the actual site? Like, is it actually a C or a T or whatever? We like don't do majority calling, which kind of is the first thing you might think, like where you look at the reads and say, oh, the most is C, so we should choose C. We actually just kind of do you pick a random read, which is kind of bad in some way because, if one read is erroneous and then you pick that random read, you have a 1 in 10 chance of getting it wrong, even though really you have, the information is strongly telling you that it's the other one. But the reason we do that is because actually, if each read is slightly biased at heterozygous sites, which is sites where the individual actually has both copies, as you get more reads, the bias builds up. So it's definitely something we think about. And I mean, I don't want to say that like I don't want anyone to come away from this thinking that like the field is fake. I mean, the field is really robust. And it's actually incredible the insights we can make. But definitely, it's always suspicion and skepticism are often really valuable.
Yeah, and like when you're on the cutting edge of something, when you're on the cutting edge of a science or a new, set of methodologies, people actually don't know often what type of errors are, very common. And so people learn from the mistakes that they make. And so papers like yours are important because, the whole idea is other people read the papers and they know to be careful of certain things. And they are careful of certain things right now, in terms of, like, for example, and some of the listeners know, some of the early detection of natural selection in certain populations turned out to be just population structure artifacts. And people are very, very aware about that right now. They were not super aware of that, a decade ago, but now they are. So, it's a learning process. So can you talk about, this particular paper about the Red Deer Cave individuals, like what the problems specifically were that you saw and how that impacted the conclusions that you could or couldn't draw?
Yeah. So I guess the first thing I noticed was that like I didn't think the conclusions were super well supported. I thought they were just sort of the samples also has low coverage, so a lack of power. And then I started to like look into the data more.
So just to be clear,
Okay.
Tell them what low coverage is.
It's a good point. Yeah. So basically, if you were to look at the average position in the genome and say how many times do we have a read that's overlapping this position, if you were to have 30x coverage, that means on average there's 30 reads that overlap, like 30 sequences of DNA that overlap the that position. Which then gives you like very high confidence what the position actually is. You can even like do what's called diploid calling and get both reads because humans have two copies, one for their mom, for their dad. But this individual had like 0.03x coverage, which is quite low. I mean, there's definitely lower, but like in our field with ancient DNA, you often don't get 30x coverage, which is maybe like a modern genome. You might get like 2x coverage or like 0.5x coverage, but 0.03 is like quite low. And so like they were saying that some z-scores, which are sort of like a measure of significance with some like questions they were asking, were not significant, which is interesting. But like it's not clear. It wasn't clear to me that this was because the answer was like neutral. It's sort of the question they're asking is MZR closer to population A or B? And they said, well, we can't tell. And is that because that they're equally close to both, or is it because of a lack of coverage? So then I started going into the BAM more carefully and noticed like a number of weird things about it. Like I actually don't remember the order, but like one thing is it's clearly a mixture of shotgun and 1240K sequences, and it's not clearly separated off. And then even though I then looked for contamination because it looks very much like the genome is very closely related to, or has a lot of affinity towards, I shouldn't say related to, but has a lot of affinity towards modern Han Chinese, which is like, that's like what you expect for contamination, given that this was a Chinese lab. In the same way you might expect Northern European contamination from like a lab in Germany or like, a sample from Germany, which might be a German archaeologist, dug up or something. So I thought maybe it was contaminated. But actually it does show the typical pattern of like damage, which you should expect, which actually was a little bit surprising to me. And then, I actually think it's very interesting that the claim was true. But I kept looking at it, and actually the pattern of damage, which of like C to T damage at the end of the read, was also present for like C to G or C to A or A to G or A to T or anything else. Like all of the sort of subcategories of like possible mutations were all extremely enriched at the ends of the reads, which is not what you expect. And it seems to indicate that some other process was potentially generating that C to T profile, which the authors had looked at and said, oh, well, it doesn't look contaminated. And you even can restrict to sites that have evidence of damage, which is like, there's some samples that are like really amazing and interesting, but like unfortunately, because that it's really easy to contaminate them, they have contamination. What you can do is you can just restrict only to reads that are clearly damaged. And so they like looked at this and they said, oh, it looks super damaged. And actually they did like a thing that like checks the proportion of damaged reads and it was super high. And so they said, no contamination at all. But yeah, anyway, also I kind of am jumping around and I apologize. The reason I started looking at contamination at all also was not only the Han Chinese thing, but also that the mitochondria doesn't seem to be like a single sequence, which is another. I kind of mentioned this earlier, but like another way you can look at contamination is you look at the mitochondria, which unlike the rest of your DNA, which you expect to get from two people, your mom and your dad, your mitochondria more or less only comes from your mom. There's kind of exceptions with like heteroplasmy or whatever, but more or less, 99 out of 100 times at least, you only have one mitochondria. So you can just look at that mitochondria and say, does it all look the same? Like, is there any sort of, are there any sort of mitochondrial reads, like mitochondrial DNA, which is not the same as the consensus sequence? And actually, 40% of the reads seem to not be the same as the consensus sequence, which I thought was a sign of contamination. Although, because of the high error rate, I actually sort of now I'm not even sure that the mitochondria was, or the sample, like to me the sample looks, sort of my conclusion was that it essentially looks entirely Han Chinese, so it may be entirely contamination, but sort of contamination from a single individual, which would still make the mitochondria, have a single consensus, haplotype. But it seems like something is making the reads noisy. And my best guess basically is that when you're sort of making these libraries and doing the sequencing, you'll make what's called barcodes, or like little 5 to 10, I actually don't know the exact length, sort of base reads you would kind of stick on the ends of the sequences, which then lets you kind of ID them and say if there's duplicates or not. And I think potentially that their sequencer was sequencing the barcodes and was essentially reading them as like part of the sequence, which would rapidly increase the noise, you imagine, because you're basically adding, random nonsense effectively. I mean, the barcodes are not related to human DNA. They're just sort of like random representatives of what the, some sort of like tag that's sort of orthogonal to human variation. And so it adds essentially just like pure noise at the end of the reads. I'm not sure this fully explains it, and I actually don't have a totally satisfactory answer. And despite like talking to them and talking to our wet lab people who know so much more about this than I do, we kind of couldn't figure out what the true explanation was, because there's also a elevated level of damage inside the reads, like deep inside, like 20 bases from the end, which shouldn't be caused by barcode errors. But I think barcode errors at least are probably partially explaining the thing where the ends of the reads probably are having these barcodes attached, and then they're being sequenced and treated as if they're part of the individual's DNA, when in fact they're just like a random number almost.
Yeah. So can you? Yeah. So it sounds like could be a lot of things going on here. And this is the reality of science, and what people see in the finished papers obviously is cleaned up in so many different ways. So what you're describing, I've heard in so many different seminars or lab meetings, like okay, this is like really dirty, this is messed up, like the provenance of these samples, like these machines are different in some way, or they're not, something's the library preparation, I don't know, there's all these things going on, right? But this seems like a little bit subtle, but like how did it affect the results they got and the conclusions you could draw?
Yeah, so I mean, I guess the biggest thing is just that, like, and I kind of want to say that, I forget who says, but someone said this and I liked it. Someone in the lab who was helping me with this, that like it's easier to notice there's a bug than to like find where the bug comes from. So like the clear thing is that the genome is not reliable. Like it's hard to even really make a strong conclusion about contamination, although I'm pretty confident from other reasons that it's contaminated. And like like if you look at the mitochondria, it's so noisy that like, if the same is true of the autosomes, which are like the nuclear DNA, you really shouldn't trust them. But basically the sequence just isn't reliable at all. And it should, I think it's sort of not necessarily worthwhile to think too deeply about what we can learn from this data because it's unreliable, I guess. Although my pet theory, which again is that the data is mostly coming from, potentially there is some real data in there as well, and I don't want to discount that fact. But assuming that this data is mostly from someone of Han Chinese ancestry, which is the way it looks. I want to I think I may have mentioned this in passing, but I want to clearly state that the data looks much more similar to someone of Han Chinese ancestry, even with all the noise, than it does to say the Long Lin sample, which is also actually from the Red Deer Caves, is also from the Yunnan province, a little bit younger, 12,000 years. But that is a published sequence. That one looks clean. I've actually looked into it for other reasons, and the Long Lin sample looks super clean. But as part of this project. And so it's essentially my guess is that the data is mostly contamination that has undergone some unknown process that probably related to the barcoding that generated sort of erroneous data. And so the conclusions that they made about the sort of Southeast Asia to Northeast Asia into the Americas migration were largely based on the fact that this sample looks more similar to Native Americans than a bunch of other kind of ancient Eurasians. But if you think of this sample as essentially Han Chinese or Northeast Asian, or if you just replace the sample with Han Chinese, all of the claims still happen because Han Chinese are like people of Chinese ancestry are mostly, especially those of northern China, but they're mostly Northeast Asian. They have some Southeast Asian ancestry as well, but they're mostly Northeast Asian. And so they share deeply with other Northeast Asians and Native Americans who have maybe 2/3 or so of their ancestry, maybe a bit less, but 60% of their ancestry coming from Northeast Asia. And so it's not actually the basically they weren't able to distinguish Northeast Asians and MCR in their affinity to Native Americans, but if MCR is a Northeast Asian, then that makes sense. Additionally, the idea that MCR is closer to Native Americans than other Southeast Asians or to West Eurasians also makes sense because Northeast Asians broadly are like that. So it's sort of this alternative hypothesis that I sort of raise is that the data is mostly Han Chinese or Northeast Asian in origin. It sort of explains the main findings of the paper in a way that don't require you to invoke the sort of novel migration, which, while very interesting and even potentially real, I mean, I don't want to discount the fact, again, there is this population-Y signal, which is kind of interesting and confusing and maybe one of the biggest mysteries in ancient DNA. But so maybe there's something there, but I don't think paper is good evidence for it, I suppose.
Yeah. And so, I mean, I think it's a good segue, moving from the issue with, all the method sections, which are super important, but maybe not as interesting to the layperson as the result sections. And so you talk about population Y, which, I talked to Pontus Skoglund, who was in, your lab a decade ago about this
as well, right?
Yeah, he was, and he thought, so they initially, from what I recall Pontus telling me was they checked over and over again because he thought it was some sort of error. It was so strange. And, I actually was assuming for the first year or two that people would have a follow-up paper like the one you did, where it was some sort of statistical artifact. And it has turned out not to be a statistical artifact. It turns out there is something weird going on. And so, a lot of indigenous people in South America, it's various degrees. There's some follow-up papers that show more than just the Brazilian indigenous people. But very few of those are of pure unadmixed ancestry. So the signal is probably harder to detect. So this is present. There seems to be some unexplained structure in the indigenous population of the New World. As most listeners know, New World populations mostly seem to descend from a pretty homogeneous Beringian population that expanded about 15,000 years ago. This is outside the purview of a DNA conversation, but there's a lot of circumstantial evidence that there were human populations before the arrival of the Beringians. And because of the way, the New Mexico site, White Sands, New Mexico is pretty famous in the United States, but there's also a site in Mexico that's, was discovered. It looks like they were here before 20,000 years ago, and that's probably has to be true because the last glacial maximum cut off contact. It was really difficult to imagine them coming over like 18,000 years ago, right? So there's weird things going on there in the New World. But the weirdest thing, as you said, is population Y is more closely related to southern East Eurasians, so the Andaman Islanders and maybe Australian and Papuan people, than they were to Northeast Asians. And so that doesn't make sense geographically, but that's
Also,
possibly
I want to note that it's like the Atlantic coast, as I understand it, and you can correct me, of Brazil, not the Pacific coast. So it's like which is if you actually look at Andean people and people on the other side of the Andes, where you'd think potentially like a Papuan or Australian group who like rafted over somehow might have arrived. It's not there.
Yeah, so I mean, it looks like this is old population structure, and another thing to note is this has been found in one. I think 10,000-year-old sample from Brazil that S.K. Willerslev's group looked at, but not in all of them. And so it looks like this wasn't intermixed into all of the early indigenous populations, but only some of them. We have one of them. I think that skull, unfortunately, might have been destroyed in the fire that happened like 5 years ago. Yeah,
I didn't know about that. Yeah,
we got the DNA.
Was the site, I believe. Right?
Yeah, I think so. Yeah, I think it might have been in
so
that.
Unfortunate.
Yes,
it's unfortunate. That the whole thing is unfortunate.
Yeah. So you were talking about Siberia and Central Asia. It seems to me there's something we don't understand about East Eurasia and the way it was peopled, and this clean north versus south dichotomy that we see today was not as clean in the past, because I still think it's much more plausible that population-wise ancestors came over Beringia somehow, perhaps through a coastal route, or perhaps through a structure in Siberia that we don't understand. We also have a very, very old substrate, it looks like in Tibetans, which I'm sure you know about, and also Tibetans, I believe, have haplogroup D, which is also found in the Angi. Most Tibetan ancestry is from, Yellow River, farmers, but that's recent.
Did you see? I'm not sure. Maybe you're referencing it or not, because there's some idea of this beforehand. But did you see the recent paper by Tianyi Wang, out of curiosity, on this?
Give me the summary.
Basically, there's this sample also from Yunnan, which is where Red Deer Cave is. That's quite old. I forget the age, but it looks like a sort of in the I think you're kind of right about the way you're describing the branching of Southeast Asians, where there's like a ton of diversity, and like it's not actually like for example, people don't know this, but actually there's not, like, if you compare South Indians to like Onge and like Vietnamese or something, actually they're no closer to Onge than they are to Vietnamese, or vice versa. Like there's just a huge amount of diversity. Like, so like the ancient South Indian ancestry, which is not necessarily Onge, is like its own like deeply diverse Southeast Asian. Then there's the Onge, there's this newly described one. Tianwan is obviously Northeast Asia, but it kind of like so there's just this like branching of these diverse clades where the branching order is not clear. And anyway, so they discovered another one of these. This I forget the name of the sample. I believe it starts with a Y, but anyway, it's from Yunnan. This divergent Southeast Asian ancestry, and interestingly, it seems to be ancestral to Tibetans. It is the Tibetan ghost, and also it's ancestral to Wang Lin, which is the sample I mentioned a little while ago.
Yes, I think I did see that. Yeah.
It helps clean up this sort of complicated thing where, I mean, my view is sort of that is just like amazing diversity there, probably because it was a wonderful place if you're an early human in the Pleistocene. Like Siberia is covered in ice and it's full of mean mammoths, but if you're in the jungles of Southeast Asia, you're gonna have a wonderful time and you can diversify a lot more.
Well, so what we see, what we see now is like the initial upper Paleolithic. We see a rapid out of Africa, or maybe out of the Middle East, but whatever population absorbed the Neanderthal admixture out of perhaps West Asia about 50,000 years ago, in that range, expands eastward, right, and diversifies really quickly. And that's what you're talking about, right? And then there was, now there's a model, and I don't know if you accept this or what the Reich Lab thinks about it. There's a model that there's a second wave that comes out of West Asia that is the ancestry of most West Eurasians, that replaced the earlier settlers in Europe around like 38,000 years ago. Like, it's a hub theory, right?
Yeah, I mean, I don't think it's wrong, but I wouldn't I don't strongly think it's right either, I guess. I mean, I'm sort of agnostic. I think it's interesting. I think it makes sense and it follows the data. I mean, what's clear is that like the people of Europe, who we have like pretty good samples for. I mean, the first people of Europe actually aren't even related to either group. I mean, like the Zlati Kun individual, who actually is like sort of deeper split than like East or West Eurasians are from each other.
Yeah.
But then the next kind of set of early, Europeans, this individual called from Bacho Kiro, another one from Oase, they're not really super closely related to modern day East Asians, but they clearly are on that branch. They're like early splitting East Eurasian, while actually later samples from even those same sites, at least I think Bacho Kiro, maybe also Oase, we have later sites, later samples. Those are clearly related to West Eurasians. Like the closest ancestors are for the modern people of Europe and the Middle East. So it's not clear where they're coming from, and I don't want to make a strong claim about the paths.
Sure.
Although I think the hub theory that they're kind of hiding out in the Middle East makes total sense. Like maybe Iran, I think, is where they claimed it. Paper correctly. And it makes total sense. And certainly there was a replacement event. But I think the geography is Iran is a sensible place, I guess, but who knows? Maybe they're still in North Africa. Maybe they're hiding in. It's sort of you kind of somewhat reference it as well. But obviously, all modern humans have recent ancestry in Africa in the last 60 or 70,000 years. But we actually don't know where the out of Africa bottleneck happened, whether it happened in Africa as they were leaving Africa and somewhere in southern Arabia. We don't know how long they were hanging out in Africa or southern Arabia before they spread into the rest of Eurasia. There's a lot of unknowns here, basically.
Yeah, I talked to your advisor once about this a decade ago, and it's we talk about this out of Africa bottlenecks, this out of Africa population, and there were some early papers using whole genome data to do the plots, do the PSMC or MSMC, whatever, reconstruct the population growth and all that stuff. We got that, right? But there hasn't been a lot of detailed exploration of that period, partly because we don't have ancient DNA. And it's just
get it from that period. And also from North Africa or East Africa or South Arabia. Those are tough places to get the data from in general.
Yeah. Sunday, who knows? They're dry, but so there's a lot of lack of clarity of this early but essential period. And so, we actually had an exchange. I don't know if it was public or in DMs because I was curious. So now we have so we have this early East Eurasian expansion. Possibly we have another branch later on out of Iran, but let's just bracket these together. They're the people that have that Neanderthal admixture pulse, the dominant one. But then we have Basal Eurasians that expanded maybe 10,000 years ago. And we know Basal Eurasians, this group that's not sub-Saharan African, they went through the bottleneck. We know that, right?
Yes.
So they went through the bottleneck, but they didn't get as much of the Neanderthal ancestry, if at all.
Yeah, there's not samples for them, but given the you can kind of make a correlation between the amount of Basal Eurasian ancestry, both in ancient and modern samples, which I think is as early as 24,000. Zuzuana maybe is the first one we see that really has
it.
But anyway, basically you can correlate percent basal and amount of Neanderthal, and it looks like it's zero essentially. So it's none at all.
Yeah, and I know that Laurent Scoffier's group had a paper where they had a model that required no Basal Eurasian, but I haven't seen any follow-ups on that. So it looks
I haven't seen that. I should. They do very interesting mathematical things. I like that. I like their papers.
Well, I will tell you a funny story about that because I'm old. I read a Scoffier et al. Paper about how it was mathematically impossible for there to be Neanderthal admixture in 2006. And then after it was discovered, they figured out how it's mathematically possible. Jeff Wall is pretty annoyed by that because his paper that showed Neanderthal admixture from 2006 still gets less citation than the other paper.
Oh, that's unfortunate. I people should cite Jeff Wall. I've actually not read that paper, and I have read the Scoffier paper. I think I have. I thought it was from 2003, but there's that paper actually is interesting. It's the range expansion one, and I think actually what it's telling us is not that there was none. Obviously, that's not true, but the rate of it actually may be saying something about the hybrid incompatibility. Actually,
well, there was hybrid incompatibility. I don't want to get into that because that'll be a little off topic. But then we have ancient North Africans that were recently discovered.
This is Takakuri you're talking about.
Yeah. So that's so they split off even before the Basal Eurasians, and it's still unclear from what you have told me, and you would know, where in your circle people would know. It's still unclear whether they went through the bottleneck. So maybe they didn't go through the bottleneck, and so the bottleneck was later. I don't know.
Yeah, I actually think it's even not totally they're definitely not just Basal Eurasian. Actually, there's something there's that sample is so amazing and so fascinating. That's such a good paper, and people should read it. It's and people should play with the data. The data is a diamond. It's really amazing. Even though, unfortunately, as I'll kind of come to, it's very low coverage. But it's clearly it's closer to Eurasians, even though seems that only has a drop of West Eurasian backflow, where like modern or like later North Africans have a lot more West Eurasian backflow.
Yeah.
It has almost no Eurasian ancestry, and yet like it's closer to Eurasians than like any African who they themselves don't have major Eurasian ancestries. Like so Somalis, for example, are closer to Eurasians than Tapu Koreans, but that's because Somalis have like 60% of their ancestry or something, maybe 40%. I'm forgetting the number, but like around half of their ancestry coming from West Eurasia. Anyway, so the point is, and that meant I apologize, the number's wrong. But the point is that like if you look at a person who does not have Eurasian ancestry and Tapu Kori will always be, even ones who have like a little bit of Eurasian ancestry, like a similar like 10% amount, Tapu Kori is like clearly a clade of Eurasia in that way. But the Mota individual, who's like kind of the previous winner for like closest to Eurasia without being Eurasia, like actually if you involve them, it looks like there's some ancestry in Tapu Kori which is like deeper than Mota. So like that clearly is
okay.
Mixtures. I think Tapu Kori, like North Africa and the Sahel and like the Green Sahara, I mean, there's a lot to be done there. There's a lot of interesting stuff there. Anyway, the thing I want to say is I think it's not clear. They might be the Basal Eurasian. They might be partially Basal Eurasian. They may not be the Basal Eurasian. That's sort of just my view. And then when it comes to the population size and the bottleneck, unfortunately, because it's low coverage, you kind of need, I mean, I can go into this if people want, but to use PSMC, which is the tool that people use to infer population size back in time, you need diploid data, which is
because it's patterns of heterozygosity.
Yes, exactly, and you will read hets, and basically to do that, I kind of mentioned before, you need like 30x coverage, and I believe Tapu Kori is like 0.05 or something. I mean, it's really tragically low for how amazing it is, but it's actually still so much. It's so powerful, that tiny drop of data, that like you can learn a ton of things. Like I think that's a great.
Really quickly for the listener, some of the listeners are engineers and mathematicians, so they think technically. Just to be clear, what we're talking about here with PSMC and these population sizes and heterozygosity, it's like you have a genome is 3 billion base pairs. It has like, maybe 4.5, 5 million single nucleotide polymorphisms. That's the variation you're working with. The pattern of that variation, heterozygosity, is like the two different copies of the genome have different bases, different states. The pattern of that variation along the genome is affected by population history. And so we can now look at that pattern of variation at really high precision, and from that, test models of various population history that could result in that pattern of variation. Now, if you have low coverage data, you can't get that pattern of variation to the level of precision to make a really good, inference about the population models that might fit that variation, right?
Yeah, exactly. And just to add on to what you're saying, and if this is too technical, please stop me. The way PSMC basically works is, at older events, because of recombination, are like smaller. So like the chunks of Neanderthal ancestry in Eurasian are very tiny. I mean, like there's lots of them, but they're like little slivers. Versus if you look at someone who's, let's say, Latin American and has a mixture of African, European, and Native American ancestry, those are long chunks because the events are, or at least compared to the Neanderthal, because it's relatively recent. So what you do is you look across the genome and you kind of you make like, this is a little bit of a simplification, but you kind of look at the genome and kind of paint it by like amount of heterozygosity. So it's like, oh, there's a lot in this kind of bunch, and there's like a little bit here, and then there's a lot, and there's a little bit, and you kind of see how long those things are. So like you like have like a chunk that's like a certain length and is a certain amount heterozygous, and then you switch to a different like amount of heterozygosity, and you have to kind of make a choice, like how much heterozygosity there is in a genome. You can't it can't be a gradient. You're kind of like blocking it in some way. Again, this is kind of a simplification, but once you have that, you can then kind of look at your layout that you've built of like how much heterozygosity there is at different chunks and say, well, these chunks are a certain length, so they're from like this period. It's actually kind of more the other way around. You actually really look like changing time periods and then looking at the heterozygosity in those time periods. But effectively what you're doing is you're getting like a map of thanks to the fact that older events are more chopped up, that like if parts of the genome that are more chopped up are more heterozygous, then like there's a big population back in time. But if these like chopped up things are actually less heterozygous, then there's a small population back in time, because the amount of diversity, which is kind of the amount of heterozygosity, is like it's like sort of related to the population size. And then long chunks, same thing. So if long chunks have more heterozygosity, then there's like a big population recently in time. And if there's a big chunks have small amounts of heterozygosity, then it's a small population in recent time.
Yeah, and I want to move back to population history and stuff, but really quickly, technically, I want the listeners to understand this sort of logic of just like looking at patterns of variation, heterozygosity, SNP diversity, all of these statistics. Looking at these is a very old way of trying to understand population history and also like the impact of stuff like adaptation, natural selection. For those, old people out there, there's things like Tejima's D, which uses, simpler pre-genomic data, but usually across species comparisons and looking at heterozygosity and SNP diversity and comparing these statistics. So, this is how the sausage is made. We'll give you a little bit of a window into that. But back, before we finish up, I want to talk about East Eurasia and Siberia and Central Asia and stuff like that. There's a lot of papers coming out, especially out of China. Obviously, Qiumei Fu, who's a alum of
She's
your
amazing.
lab. Yeah,
I don't know. She's so powerful. Yeah, that's all I'll say. She's awesome.
yeah. So there's a lot of work coming out about this part of the world. With the recent time periods, I think we have more or less some clarity now of okay, there's the Yellow River farmers, there's the Yangtze farmers, there's this deep lineage in Southeast Asia that split off earlier, might be close to Australasians and whatnot. There's two questions I want to ask you, and you might not know, or have a strong opinion. One, an older layer than that initial Upper Paleolithic. I think Estonian group, Mate Mapsalu and Luca Pagani, some of them have found this in the Papuan data. I know people in your lab have decade ago told me they thought that wasn't a real signal, that it was just the issue with the assembly of the genome is actually causing the problem. So when you're looking at these supposedly looking at weak signals from not super differentiated populations, it seems like the contamination issues and the way you assemble the data, these methodological issues loom large. And the second issue is just in terms of how much the population structure, say, 30,000, 40,000 years ago when you had Tianyun Man and all these, are reflective of what we see in, say, the Holocene, where we seem to see pretty intelligible north-south variations. So, in Southeast Asia, until 4,000 years ago, much of Southeast Asia was dominated by people more like the Onge, right? And then there was the rice farmers that are coming out of southern China, Austroasiatic speakers and whatnot. And now we kind of see a pattern that we understand based on that. Can you address both those issues, like the older, possibly, pre 50K populations, and there, this is a lot of it driven by Australia, but as well as these old, I think artistic representations. I think in
The
Borneo.
new things that are Indonesia. There's a new.
Yeah,
It's really
yeah.
amazing. It's pretty amazing.
Yeah. And then the second issue would be just the structure, say 40, 30,000 years ago in East Eurasia and how it relates to today.
Yeah, sure. No, I'm happy to tackle both. Basically, I want to start off by saying that the divergence between this sort of question of did Papuans diverge first from the rest of Eurasians and then kind of intermix with East Eurasians after to make them seem a little bit closer, or were they always closer to East Eurasians, and maybe actually the thing that's driving the signal is that the Denisovan ancestry they have kind of pulling them away. And I always want to say there's someone actually in the lab now who I guess I won't name names, but I don't think you would mind anyway. Regardless, this person believes that the Papuans are the first split. David still believes Papuans are a clade without the Denisovan ancestry with East Eurasians. So even in the lab, I don't think people fully agree because it's an early branch and it's tough to say. I lean more towards the camp that David's in, and I think there's good evidence for both. I mean, it's sort of also it's somewhat maybe it's a philosophical argument because people are a mosaic of ancestries. Your ancestors on your chromosome 1 are not the same people as the ancestors on chromosome 2. So obviously Papuans have some ancestors deeper than Eurasians in the Denisovans, and how you want to frame it, and if there's admixture from East Asians, what does that mean? Getting into, I'm kind of actually answering it in 3 chunks, but hopefully this answers your 2 questions. But getting into those old samples from, or the old cave paintings from Indonesia, which I think are 70,000 years old, and I don't have a reason to doubt the date. Not that it's my expertise, but I believe that the date is about that. Maybe it's 60 something, but very very old, and evidence for early human habitation in Australia as old as 60,000. I'm not going to comment this from an archaeological perspective, but clearly the answer there is whatever. If those dates are to believed, and I see no reason why they shouldn't be, they either must have been early humans who were replaced by later humans in much the same way that we see in Europe, which would not be shocking. I mean, it's actually like, again, in Europe, before we see people who are even remotely related to modern Europeans, they still weren't probably that ancestral. Like the Curvatians don't seem to contribute that much to modern, it's like a little bit, but it's really a tiny sliver of modern European ancestry. But even before we see these people who are actually European related, we see at least 2 waves of people who are totally unrelated. And so, like, it's possible that there was ancient humans, Homo sapiens related people, who were living in ancient Southeast Asia, ancient Australia, 60,000 years ago. If that's the case, though, we know that they can't be ancestral because that we can see that the divergence times between all Eurasians are like on the order of like 40,000 years ago, and we can date the Neanderthal ancestry, which is shared by all Eurasians, although again there's some depletion in people with more Basal Eurasian ancestry, which specifically is kind of Near Easterners. But anyway, other than that sort of depletion, all Eurasians share this Neanderthal ancestry, which is dated to I think like 45,000 years ago. And there's a recent paper in the last year, which actually 2, I think, that came out kind of together that like dated really confidently. And so like clearly, whatever these early people are, they were replaced. They may not have been human though, because like we don't really understand Denisovans that well. I mean, we just really in this same last year, which is amazing. It's been an amazing year looking back on it. We got confirmation of like a morphological skull being the Harbin skull being a Denisovan. But like the in general, like before the emergence of Homo sapiens and Neanderthals, like the Afro-Eurasian or West Eurasian and African records are kind of really messy. It's this whole thing called the muddle of the middle, and the East Asian like archaeological and like morphological record, as I understand it, are quite messy. Like there's all these skulls which kind of look like some parts of them are super modern looking, like modern Homo sapiens. Some of them look really Neanderthal-like. Some of them look super like Homo erectus-like. I just think it's not totally clear what's happening, especially in Southeast Asia. And hopefully we can clarify this. But like potentially, Denisovans are making art, and I don't see a strong reason why that can't be the case either. So like there is evidence, as you were saying, of these like early people doing all sorts of interesting things, 60,000 plus years ago, and I have seen people kind of debate the dates, and maybe there's good reason to, but I have not been convinced that the dates are wrong. But clearly these people were doing art and things like that, but they were not necessarily they were not, surely were not the ancestors of the modern people there, whether they were human or Denisovan, or, maybe Denisovans are humans, whether they were sapiens or Denisovans. It's sort of, hard to tell. And then to your final question, I mean, like, there's so much that can go wrong for your tribe if you're living in the Paleolithic, as I see it. I mean, I'm not like an expert on this, but like, like you could get wiped out by a disease, you could all starve to death, you could be murdered by the neighboring group. And so if you look at the like groups who contribute to modern people, they're like a sliver of the diversity that used to be there, especially in East Asia. But even in West Eurasia, I just mentioned these Curvatians, the Orgnatians, all these cultures which are existing, which, like ancient North Eurasians definitely contribute to some degree to like Native Americans, especially, but in Europeans, but like they essentially have been like, there's no one on Earth more than like 40% ancestry and of people in Eurasia. There's like, I think the max is like maybe like 30 something percent. I mean, like, a lot of people who used to exist no longer exist in like especially in an unmixed form, and some people don't exist at all. Like Tianwan Man, and I don't think it's super clear that like that individual has like that population, which we have actually 2 individuals from. There's also this 33,000-year-old sample from the Amur River region of China. So also northern China. And like, it's not clear that they contribute to later groups. And so like, maybe the question you're asking, I'm not sure, and I apologize, I'm not answering it properly, is like, where are these groups emerging from? Like, what happens to the other groups? And I think, for some we kind of have like, for some, like Duzuana, like very clearly is like contributing. That's like this 24,000-year-old sample from the Caucasus I mentioned before. It's clearly contributing to later West Eurasians. Like it's basically the same ancestry you see in the Anatolian Neolithic, which obviously is like a major contributor to farming in West Eurasia, and so it's like common across West Eurasia now. And so like for some groups we have this. West Eurasia is sort of better sampled because like historical reasons, I guess. But hopefully we can sample the entire world really deeply going forward. But anyway, basically, it's not clear where these groups are hiding out, and What happened to Tianwan Man? Did he contribute anything at all? It's sort of hard to say without getting the the main component of modern East Asian ancestry. Like if we were to find someone who is like the main contributor for modern East Asians, we might find actually it's mostly that mystery person, but actually the Tianwan individual is also required. You also need like 10% Tianwan like ancestry. So I don't know if that exactly answers your question, but I just think that like if you look at the record, you can tell that like human groups turn over all the time, often by groups that are very similar to them, although occasionally by ones that are very, very different. In the case of maybe Neanderthals and Homo sapiens, but this sort of turnover is so high that actually if you look at like any given ancient group, it's like not clear that they have any contribution to modern groups at all, especially in deep.
Yeah. Well, I mean, that was one of the, talking about ancient DNA and this whole field, which, has emerged like what you were probably in elementary school, maybe in middle school.
Yeah, I was like 12, I guess.
Yeah. So it's
Crazy to say.
Yeah. When you were 12, this new field that you're actually working in emerged. And one of the things with humans, I mean, obviously with other animals, it's going to be different. One of the things with humans that was a big surprise was how much turnover was. And I remember I made a prediction to Chris Stringer before the ancient DNA first came back from Paleolithic samples in Europe, that there was going to be almost no ancestry of Paleolithic people in Europe. And I turned out to be right, just because culturally we know that there was cultural turnover, and it just started to be really evident from the earlier stuff by 2015 that people just replaced each other all the time. And when you're talking about these lineages and people imagine these trees, but the issue with I think the way people need to imagine the tree is you see the branches you see the terminal branches and they go back, but like there's all these internal branches that just disappear and we don't see them. And with ancient
some of the
DNA,
also leak little bits into the later branches. It's like it's a tree, but it's like actually a crazy bush that like
it is, and over a decade ago, I remember I was at ASHG, like I think it was 12 years ago, American Society of Human Genetics conference, which is the biggest genetics conference in the country, and I remember telling your colleague Yosef Lazaridis, like looking at one of his graphics, and I'm like, "What the hell is this?" I mean, it was just like there were like arrows here and there, and it was just like, how is anyone going to make sense of it? But I mean, that's reality. I mean, reality is not simple and elegant when it comes to these sorts of demographic processes, right? And that's why it's so laborious. And a lot of the answers you've given me are very, very provisional, but that's just where the data go, right?
Yeah, I mean, I think it's important as scientists to qualify our answers. I mean, it's sort of just a philosophical opinion of mine, and I'm not saying I'm maybe a qualified answer here, but I'm not saying I'm necessarily right in that. But like, I think it's I think often people who don't do science like kind of maybe circling back, they see the claim made by the scientists for like the data and say, oh, like this is like 100% correct and will always be true, and like there's no issues here. And like I'm not saying that people shouldn't trust scientists. Like if you science is on the whole correct, and like if you read a bunch of papers from at least my field, they'll on the whole be correct. But like things are messy, life is complicated, and we often are not 100% sure about our conclusions. There are things we can be 100% sure about. Like we're now like 100% sure that humans have ancestry from Neanderthals, but like where did that admixture take place? I think that like we have some guesses, but actually we ultimately don't know. And I think it's actually, I think people in some ways would trust scientists more when they do get things wrong if they were more upfront about when they're really, really confident and when they're they're making their best guess and they're trying their best and qualified maybe what the unknowns are. So I mean that's just my own personal philosophy, which I don't know if that's the right thing to do or not. But yeah, so if I sound like I'm qualified my answers all the time, I just think it's important for scientists to do that actually.
Yeah, I mean, this is I think one of the issues here in this field in particular, like I don't want to like mislead people here and like be too strong about it, but do not trust the science because it changes, the results do change, and the models keep getting updated. And another thing that I have to say is, as is there are, many times many alternative models that can fit the data. And so you need to wait on more data before you can say which of the three models is the correct model, right?
Definitely.
And so it's just, yeah.
If I can just jump in, I just like, yeah, I mean, I think that we're sort of. I mean, I'm still in academia, so maybe I'm trying to defend academia. But I really deeply do think that, like, our enterprise is good, and you should trust us. I'm obviously biased in saying that, but I do think it's true. But, like, as you're saying, like, we're not right, but we're kind of leaning towards the truth. And I think, like, if you look at the broad, like, paintings of our field, maybe like with like Neanderthals, let's say, go back to, like at first, like, we didn't really know what Neanderthals were. Maybe they were direct descendants of like modern Europeans. Then as we kind of get to first genetic data, we realize actually, no, there's this recent out of Africa, and it kind of like becomes a new dogma at some level, a new standard that like, or the previous dogma was like, Neanderthals are the ancestors of Europeans. Then there's like a new dogma that's like, actually, no, Neanderthals aren't ancestral at all, and we all have recent ancestry in Africa. And like, then we realize, actually, no, we all do have recent ancestry in Africa, but Eurasians do have some Neanderthal DNA. They're like 2% or so snuck in, and surprisingly, it's not maximized in Europe. Actually, East Asians have a little bit, like a tiny fraction more.
Yeah, against expectation.
Than to Europeans. But so like your science has been wrong about this, and it's been corrected, and it's sort of self-corrected. But it's leading in the right direction. It's always pointing towards, like out of Africa was more correct than this multi-regionalism, but it wasn't fully correct, and now we have like a new kind of synthesis, and hopefully we'll continue to do that. I don't know if you agree how I put it, but that's
no. No, it's all about like converging on your best answer at any given period, given the data, and you update your priors based on new data. I guess like the last question I'm going to ask you then, on the way out, because this is something that's come up recently. Your mentor, your advisor, was on my friend Dorkesh Patel's podcast, actually. I think, was it a year and a half ago or a year ago?
Yeah, I remember this.
Yeah, and I'm an early guest on Dorkesh's podcast. But anyway, it's a small world. I actually hung out with Dorkesh last weekend in SF. He has, this is a sneak preview for the people that are like the paid part of the podcast. Just interviewed Elon, so watch for that.
Pretty cool.
Yeah, but so now there's a fair amount of evidence that Neanderthal admixture was higher. He gave some pretty high estimates, maybe 10 or 20% initially before there was purifying selection. And today it's like, about 2 to 3%, depending on which population, right? 2 to 2.5, something like that. I think depending on which Neanderthal reference you use, et cetera, et cetera. But also the Y chromosome and the mtDNA of Neanderthal seems to be from a deeply basal, Africanish stem human lineage.
It's closer to humans than it is to Denisovans. I mean, it's almost the best we can say, but yeah, definitely true.
And so, he said, it seems like we keep adding these, I don't think he said epicycles, but it's like we keep adding epicycles to the model. And it's like, maybe we're just like wrong in our premise somehow. And I mean, do you still get that vibe from him? I mean, do you feel that way? Like, because like some people ask me, they're like, out of Africa is like totally wrong, and this is why you have to keep adding epicycles. And I don't see it that way yet. But, I do feel like we're getting more and more data compared to, say, like, 40 years ago, and it was just like, mtDNA from placenta, Yeah.
Yeah. No, I mean, yeah, I want to first be very clear that I'm not David, and I don't want to speak for David in like that regard. Like, I can say what my opinion of his opinion is, but like it shouldn't be taken as gospel by any means. But yeah, I mean, I've seen actually this on Twitter, like people saying, "Oh, like I think often like it can be like I think it's popular among certain countries that'll kind of have like a nationalistic bent, like, oh, actually, I'm not going to say any country, but like humans came out of my country, and like
yeah, I know what you're talking about.
It's sort of in my opinion, ridiculous. But anyway, I don't mean to be too rude, but like I think that like it's like it's not really scientific in nature. It's sort of it's nationalistic rather than scientific. David he does not believe out of Africa is wrong in this sort of broad sense. I mean, out of Africa is wrong in like a pure sense. And this has been known since 2012, insofar as like Neanderthals, have ancestry. modern humans from Eurasia have ancestry from Neanderthals. I think that David is like a very skeptical person, and it's like an amazing thing. And actually, I mean, we don't have time to talk about this now. We're kind of wrapping up. But I had a whole project which totally didn't work out, looking at some interesting archaic related thing, and it had strong Z-scores, but David was like, I don't believe it, and he made me prove it, and actually I proved I was wrong, and it was a learning experience. It was good to know. But David is a very skeptical guy, and I think it's like it's a huge talent for him scientifically, and he's right to be skeptical of these epicycles. Like, the standard model of archaics, we keep just adding to it. We add, okay, there's super archaic and Denisovans, and it's Denisovans and Papuans, and now we're going to add this little admixture from ancient humans into Neanderthals, which explains the Y chromosome and mtDNA, but like, it's such a small fraction if you estimate it, and it's so noisy. Like, it kind of doesn't make sense that like both the especially like imagine like humans are patched for local, right? And the Y chromosome is flipped, is like replaced. Like, why is the mtDNA also replaced? Or imagine that they are matrilocal. Like, why is the why are both replaced? It's like a bizarre thing, especially like if you're just like imagining a 5% mixture. I mean, the chance of that happening at random is like 1 in 20 divided times 1 in 20. It's like 1 in 400. It's crazy. So like, clearly our model is kind of breaking down here, and we should be interested in throwing it out and making one anew, and maybe it'll look exactly the same as the one we already had. That's totally possible. But like, you're sort of blinding yourself at like the possible models if you're biasing yourself by trying to fit it to the old one. And like, so there's a real value to doing that. And I think that's what David was getting at. And I think that we don't really know where human ancestors, I was kind of mentioning this muddle in the middle. We don't really know where human ancestors were living like 500,000 years ago. And so I actually don't know. I watched that podcast because I was curious, but I don't remember what David says here. But like maybe humans are not living in Africa 500,000 years ago. But if people want to celebrate this as the actually they're in my country thing, and like they're not from Africa, like whatever kind of nationalistic bent, which I think is still kind of ridiculous. It's sort of a silly victory,
especially 500,000 years ago.
Yeah, exactly. Like, all humans are one. Like all human ancestors have totally coalesced at that point. Like, there's like almost no variation in human. Maybe it's like 2 million years, but still, like it goes far apple red. But like, the point is, like, all humans are one population, regardless. Like, all humans at that point, like, all of our ancestors have fully merged together. The deepest divergence between two humans groups is like 300,000 years ago. So like, whether they're living, and it's all one group. It doesn't, like, why would it matter if they came from Chile or Iceland or Vietnam or New Guinea or South Africa or Ethiopia? Like, it's all the same. But also like clearly humans were living in Africa more recently than that. Like I said, the reason out of Africa is very clear. Like, the majority of human ancestry, no matter who you are, even if you're someone from Papua New Guinea who has the most archaic ancestry in the world, your ancestors, like 90% of them or more, are living in, Africa 100,000 years ago. That's like very clear. And then like, also, if you go back like 1 million or 2 million years, then again, like, we just only have hominin fossils like at Lucy's age back in Africa again. So like, clearly Africa is a very important part of our history. Does it mean that we lived there forever? I think it's sort of simplistic and reductive, and given the amount of migration we know is happening throughout human history, like in like the Paleolithic and the Mesolithic at the Holocene. If you project that backwards, like it wouldn't surprise me if actually the majority of human ancestors were living in Eurasia. And I don't think it would surprise David either. But again, I don't want to speak for him. In like the Near East or East Asia or something, or Europe or who knows where. There are places I think we can confirm they almost surely weren't. Like, they weren't in the Americas, because we just
or Australia
there. Maybe Australia as well. I actually don't know the fossil record there at all. But yeah, there's this sort of muddle in the middle, which is very interesting, and that is sort of the time frame. It's like 300,000 to 500,000 years ago. It's exactly when this sort of interesting interactions happening between the ancestors of modern humans, the ancestors of Neanderthals, replaces the MT and the Y and the Neanderthals. And it's one of the I said that the population Y is sort of a big mystery, but this is another one. I mean, and it's something that I think. I think should be worked on. I think it's very interesting, actually.
Well, I mean, that's good for you, because you're just starting out in your career, right?
It's true. Hopefully, I mean, we'll see. I don't know.
So
I don't know if I'll solve either of these, but at a minimum, I'm excited to see them be solved, and I mean, I would love to solve them, but
for sure, man.
Yeah, we'll see.
All right. Well, it was great talking to you. I should let you go. I will see you on social media. Maybe if I come through Cambridge, let's hang out. And if you're ever in Austin, maybe at ASHG this year, maybe I'll swing by. It was great catching up with you. Your enthusiasm for the field is very, very obvious, and that's great. And I'm excited for reading your publications on Central Asia and Siberia at some point soon.
I really appreciate having me on. It was great to chat, and I would love to hang out, and let me know, and have a wonderful rest of your evening, I suppose.
Take it easy, bro.
This podcast for kids. This is my favorite part.